In DNA replication, there are proteins that help with the process, and here are their functions. In helicase, they start unwinding the double helix at the replication forks. However, ahead of time, there would be a topoisomerase, which starts relieving overwinding strain ahead of the replication forks by breaking and swiveling and rejoining the DNA strands. There's primase that starts putting down primers, so the DNA polymerase can know where to start adding the base pairs. There's DNA polymerase that starts adding new DNA strands by adding nucleotides to the DNA primer or the parental DNA. And then there's DNA ligase that starts joining the lagging strand and the leading strand with the added base sequence, so we can have a brand new strand basically.
Semi-conservative DNA replication comes from getting a double helix DNA, which is labeled as the old one / the template strand, and each little strand breaks off to create their own double helix. And when this happens, in the new double helix, one strand is made from the original / the template strand, and the new one in red is made from copying the original strand. So one strand is original and one strand is copied.
Helicase unwinds DNA. As it unwinds, DNA polymerase builds a new DNA strand from 5' to 3'. In the leading strand, since it's 5' to 3', the new DNA strand will build continuously. Unlike the lagging strand is 5' to 3'. Since DNA polymerase builds from 3' to 5', it's going to build in the opposite direction. It's going to build from back here to forward. And as it unzips, it builds from back to forward. As this happens, it will make small gaps. And these tiny little DNA fragments or DNA strands are called Okazaki fragments.
In DNA replication, DNA is anti-parallel, which means one strand will be going from 5 to 3, while the lagging strand will be going from 3 to 5. So normally, nucleotides will start replicating from 5 to 3. However, the problem starts arising when you realize that DNA polymerase only goes from 5 to 3. So that means the other strand goes straight forward continuously, such as the leading strand, and the other strand has to go in a reverse order called the lagging strand. And the reason why it's called the lagging strand is because the other strand has to start adding Okazaki fragments and starts putting the sequence in fragments, while the other one can just go continuous straight through with DNA polymerase 3.